Now let's talk about changes in oxidation state applied to a problem that is drug metabolism. So when we take a drug or a medication, say, we're putting a foreign compound into our bodies, but it's suspected that it does something to that medication. So typically you will have drug absorption, and then the drugs can get distribution, but also is metabolized. And at the end, after it does what it needs to do, the body discards that, and that goes into a process which goes to excretion. But in excretion, let's see what happens. And that's precisely the topic that we're doing when we do oxidations. So most drugs, many drugs that we take are lipophilic in nature. What that word means is that they are solubility in water is low. They are more soluble, say, in other non-polar molecules. But for the body to excrete that drug or that, say, foreign compound, what it does is oxidizes. So it undergoes oxidation. In undergoing oxidation, it becomes a molecule that is more, say, hydrophilic. And what that word means is that will be more water soluble, and that way is excreted easier excretion. So that is in general what's going on. That's why when we study oxidation in reduction, well, what's going on in drug metabolism is that a drug, a foreign compound, a antibiotic, for example, is oxidized to make it easy to excrete so that the body can discard that. But we're going to do a simple process. All of those occur mostly in liver cells. And in that liver cell, in the endoplasmic reticulum, there is a lot of enzymes that take care of that oxidation. And this is a family of enzymes called cytochrome P450. Well, we just say P450, but this is a family of enzymes that catalyzes that. So now we're going to do something and see the oxidation state. One, say metabolism, is, for example, when we take alcohol, for alcohol consumption. So in our bodies, if we take alcohol, and alcohol is ethanol, which is, let's do our line structure. That. We know that we can get intoxicated by a lot of ethanol consumption. So our body wants to eliminate that, so detoxify. So what it does in our body, well, it undergoes a series of processes, but say in the, there is an enzyme called alcohol dehydrogenases. That's one of the pathways. And these, some of those are cytochromes P450. What they do is then oxidize alcohol to another compound, and let's count the bonds to oxygen. It oxidizes to acetaldehyde. So ethanol goes to acetaldehyde. And again, in this pathway with called dehydrogenase enzymes, it can be even further oxidized to acetate. And that's even more soluble, so acetate. And let's see, what is the changes in oxidation state in that ethanol metabolism? Okay, so cytochrome P450 will then catalyze that oxidation. How we know is that it is an oxidation or not. So let's draw now all of the bonds, and we will do the changes in oxidation state. So here I'm going to draw all of that, and that is another carbon, but I am analyzing not the changes in oxidation state in this CH3, which is this one, but actually I'm analyzing the changes in oxidation state where the functional group is, so the alcohol. So I'm analyzing what is the change here. The change that we said is catalyzed by that cytochrome. Okay, I'm doing this. Now, oxidation state of that carbon. We will assign electrons in this bond to oxygen. So oxygen is going to take the 2 electrons. These 2 are going to be taken by the carbon, also these 2. And remember that because there is no difference in electronegativity between these 2 carbons, these electrons are split in half. So when we do the oxidation state of this carbon in the box, we're going to say 4 valence electrons minus, and then we count how many electrons are assigned. So we say 2, 4, 5. So it is -1. What happens here? This is acetaldehyde. So assigning electrons in that carbon in the box. So the 4 electrons here go to oxygen, but this go to carbon, right? Because carbon is more electronegative than hydrogen. And here, 2 carbons, we split that in half. So we say oxidation state is 4 valence electrons, and then we count 2 electrons here and 1 more, that's 3. So that's a +1 oxidation state. Notice oxidation state had increased. That is an oxidation. And lastly, let's see the last change. Here, oxidation state 4 valence electrons, and then those 4 electrons are assigned to oxygen. Also, these 2 electrons. And this, remember, this is a CH3, so that was another carbon. That's why we split it in half, and we say 4 valence electrons minus only 1. And that is a +3 oxidation state. And again, we see that this is an oxidation. But again, many mechanisms like that are happening in our bodies to detoxify, to metabolize drugs.